All right, if you've got a biochemistry exam on the horizon, then you are in exactly the right place. We are about to do a deep dive into the one element that literally forms the scaffolding for all life as we know it, and that, of course, is carbon.
So let's just jump right into the big question. Why carbon? I mean, of all the elements on the periodic table, what is it that gives this one atom the unique, almost magical ability to build the huge, complex molecules that make life, well, life? Let's break it all down.
Here's our game plan for this review session. We're going to start with the absolute fundamentals of why carbon is the star of the show. Then we'll take a little trip through its history and structure, and we'll wrap it all up by seeing how it assembles the massive molecules that define biology.
Just think about it for a second. From the tiniest little bacterium to the biggest blue whale, the architectural blueprint for all of it starts with carbon's just incredible versatility. Its ability to build these large, complex, and really diverse molecules is the fundamental reason life on Earth is so incredibly rich and varied.
And you know what? It all boils down to this one magic number, 4. Carbon has 4 electrons in its outer shell, its valence shell. This makes it perfectly balanced. It doesn't really want to give away electrons, and it doesn't want to aggressively rip them off from other atoms. No, carbon is the ultimate sharer in the atomic world.
So the really crucial point here is to understand how this atomic setup translates into a biological superpower. It's one thing to say it has 4 electrons, but it's what carbon does with them that changes literally everything.
And here's how it pulls it off. Those 4 electrons allow carbon to form 4 incredibly strong covalent bonds, essentially turning itself into a central hub. And this isn't some flat, boring structure. No, it creates a 3-dimensional tetrahedron, kind of like a little pyramid. And that 3D shape is the absolute key to building the complex architecture of life.
Okay, let's take a quick detour into the history books to see how we even figured all this stuff out. It wasn't obvious at all. In fact, understanding carbon's role sparked a huge scientific revolution that completely changed how we think about life itself.
For a really long time, scientists believed in this idea of vitalism. They thought that the molecules made by living things had some kind of mystical life force in them. But then, in 1828, a guy named Friedrich Euler accidentally made urea, an organic compound, in his lab, and that idea just shattered. Fast forward over a century to 1953, and the famous Miller-Urey experiment showed that amino acids, the building blocks of proteins, could just form on their own in early Earth-like conditions. That was the nail in the coffin. The chemistry of life follows the same rules as all other chemistry.
So all that brings us to what we call the carbon skeleton. The best way to think about this is like the frame or the chassis of a car. It's the basic framework that you can then build an incredible variety of different molecules on top of.
And this is where the real artistry begins. All of that molecular diversity comes from 4 main variations. You can change the length of the chain, making it short or incredibly long. You can have straight chains, or you can add branches. You can change the position of double bonds, which totally alters a molecule's shape and properties. And finally, you can have the skeletons loop back on themselves to form rings like we see in sugars and steroids. It's like a massive chemical library with endless possibilities.
This incredible variety leads to something called isomers. These are molecules with the exact same chemical formula, but different structures. Think of it like a set of Legos. You have the same number and color of bricks, but you can build them in different ways. Structural isomers are when you just connect the bricks in a different order. Cis-trans isomers are a bit more subtle. They're about how the pieces are arranged in space around a double bond. And that tiny difference in arrangement can mean a huge difference in how the molecule actually works.
And then you have enantiomers. The easiest way to think about these is to look at your hands. They're perfect mirror images of each other, right? But you can't superimpose them. That's what enantiomers are. And this is so important in biology. For instance, your cells almost exclusively use the left-handed versions of amino acids. It's also why one mirror image version of a drug might save your life, while the other version could be useless or even harmful.
Okay, listen up, because if you're studying for a test, this is the part you really need to drill down on. If the carbon skeleton is the frame of the car, then functional groups are the engine, the wheels, the steering. They're all the parts that actually do something. They're specific clusters of atoms that attach to the skeleton and give each molecule its unique chemical personality.
Think of this table as your ultimate cheat sheet. You've got the hydroxyl group, which makes things polar and soluble in water. It's why sugar dissolves in your coffee. You have carboxyl groups, which act as acids, and amino groups, which act as bases. You stick those 2 on the same molecule, and boom, you've got an amino acid. The sulfhydryl group is like molecular Velcro. It forms these crosslinks that are critical for holding proteins together. And then the methyl group, it's like a tiny switch on your DNA that can turn genes on or off.
Each one of these groups gives the carbon skeleton a specific job to do.
And to really hammer home how important these are, just look at the phosphate group. Whenever you see a phosphate group, I want you to think one word: energy. This group is the key to ATP, the molecule that is the energy currency for pretty much everything your cells do. Without this one functional group, the entire energy economy of life would just grind to a halt.
So, we've got our carbon skeletons, we've got our functional groups attached to them. Now for the final piece of the puzzle. How does nature use all this to build the giant macromolecules of life, like proteins, carbohydrates, and DNA?
It's actually shockingly simple and elegant. All of life's giant molecules are built and broken down using two opposite reactions. To build a long chain, a polymer, you link the smaller units together by removing a molecule of water. That's a dehydration reaction. To break that same chain back down, you just do the reverse. You add a water molecule to break the bond. That's hydrolysis. So just remember, water out to build, water in to break. Simple as that.
And that leaves us with this final thought to chew on. We've seen how a few simple rules, carbon's 4 bonds, a handful of functional groups, and this simple build and break mechanism are all it takes to generate the breathtaking complexity of all life on Earth. It really makes you wonder, if these are just the basic building blocks, what else could chemistry be capable of creating? What are the ultimate limits to what it can build?